One of the biggest questions facing early geneticists was how DNA was used to encode for proteins. Many scientists worked to determine which codon sequences encoded for which amino acids. And in 1961, Marshall W. Nirenberg and J. Heinrich Matthaei became the first scientists to match a specific codon sequence to an amino acid. They did this by radiolabeling a single amino acid and then running the experiment 20 times, once for each amino acid, to determine which formed proteins containing the radiolabeled amino acids. The first to be done was the sequence of repeating U's, or poly-U mRNA. When phenylalanine was radiolabeled, a newly synthesized protein was formed containing the radiolabeled amino acid. This led to the conclusion that the UUU codon encodes the amino acid phenylalanine, though at the time the three-letter codon had not yet been proposed. This was also used to determine that AAA encodes lysine and CCC encodes proline. However, this experiment did not work for the GGG codon. Nirenberg continued to work on the genetic code with the scientist Philip Leder, and in 1966 they deciphered the rest of the universal genetic code used today. They did this by putting specific trinucleotides in a mixture with ribosomes and 20 different tRNAs. However, one of the tRNAs contained a radiolabeled amino acid. They then allowed binding to occur, and the unbound tRNAs, along with their amino acids, were filtered out, and the filter paper was checked for radioactivity. When the filter was found to be radioactive, this told Leder and Nirenberg that the three-letter sequence that they had included in the mixture was what encoded for the radiolabeled amino acid. This resulted in 61 codons being identified, and the last codons were later determined to be stopped codons.